Hello everyone. In this video, we are going to discuss some of the properties of mature RBCs. So RBCs have certain unique properties and we need to understand them because when we discuss about anemias, there are some abnormalities in this normal characteristics of RBCs that will result in various types of anemias. So these basics are very important to understand. The first thing is what is the shape of the normal mature RBC? So shape of normal mature RBC is biconcave shape. So it's a biconcave shape structure as you can see here. Now why this is biconcave? Because RBC has a bigger surface area, but the content inside RBC. So RBC is filled with hemoglobin. It is filled with hemoglobin. The hemoglobin content inside the RBCs are less as compared to its surface area. So they have a bigger surface area, a bigger volume, and the amount of hemoglobin is less. That is why the entire structure of RBC, it is not completely full. It is not full. So that is why the RBC, they take the shape of a biconcave disc. And if you see it under the microscope, you can see something like this. You can see something like this. So basically this hemoglobin will take up the peripheral areas, and the central part of the RBC, will remain free of hemoglobins. So this is how a normal RBC looks like. Okay, so it will have a central pallor. So central pallor, it is a characteristic of normal RBCs. Okay, and this is the electron microscopic image, and you can clearly appreciate the biconcave shaped RBCs. Now, because they have increased surface area, it has certain advantages. Okay. So because of increased surface area, as we know, the normal function of RBCs is to exchange the gases. So they carry the oxygen and carbon dioxide and they help in exchange of gases. And because of this increased surface area, the gas exchange happens in an efficient manner. So exchange of gases, you can say efficient, very efficient exchange of gases happens because of this increased surface area. Okay. And the second property is this increased extra surface area will allow the RBCs to become more flexible. So it increases the flexibility also. Increased flexibility. Okay, so these are the two important consequences of having extra surface area. So there will be efficient exchange of gases, those are carbon dioxide and oxygen, and it will make the RBCs more flexible. Now, why this flexibility is important? Because we'll also discuss that RBCs, when they circulate through the spleen, especially the open circulation of the spleen, they tend to negotiate through very narrow slits in the capillaries. Okay, they tend to negotiate in very narrow slits in the capillaries. And if they are not flexible, they will get caught up in the capillaries, and after that they can be destroyed by the macrophages. Okay, and to avoid these things, the RBCs are highly flexible so that they can pass through the narrow capillaries, and that is how they can survive in those conditions. Okay, so these are the consequences of having this increased surface area, which is responsible for giving this biconcave appearance. Now we will understand what are the factors which maintain the integrity of this cell membrane. So RBC cell membrane, like any other cell membrane, it is a phospholipid bilayer, and the integrity is maintained by three important factors. The integrity is maintained by three important factors. The most important factor is the cytoskeletal proteins or the cytoskeleton. The cytoskeleton is the most important factor which helps in maintaining the integrity of the RBC membrane. We'll discuss more on this when we will discuss about the hemolytic anemias caused by RBC membrane alteration, for example, hereditary spherocytosis. We'll discuss more on RBC cytoskeletons. Apart from cytoskeleton, the electrolyte and water content of the RBC also helps in maintaining the integrity of the membrane. And the amount of lipid. Okay, so the lipid content also is a determinant of cell membrane integrity. Okay, so these are the three factors which helps maintaining the cell membrane. But the most important factor that you have to remember is the the cytoskeleton. Okay, the cytoskeleton. Okay. So this is about the shape of the normal RBCs. Now we'll see some more properties. Now a mature RBC, it lacks mitochondria. So mature RBC lacks mitochondria. And it also lacks a nucleus. So mature RBC doesn't contain nucleus. Okay, so they do not contain the nucleus. Because they do not have nucleus, they cannot synthesize DNA and RNA. So nuclear materials they cannot synthesize. So DNA and RNA are not present. These are not present. So that's why if you see a normal RBC, as you can see here, this is a normal RBC. It doesn't have a nucleus. Nucleus always takes the blue color. Nucleus contains DNA and RNA, and DNA and RNA they are basophilic, so they will take out the blue color. And you can see the normal RBCs, they do not have a nucleus. Sometimes in the peripheral blood, you may get this kind of RBC. These are known as NRBCs or nucleated RBCs. Okay, NRBCs and or nucleated RBCs. These are important because normally you will not find nucleated RBCs. So if you are finding nucleated RBC, that means there is some abnormality. This usually happens whenever there is severe hemolysis. Okay, so whenever there is severe hemolysis, the precursor forms of RBCs from the bone marrow will come to the periphery. Okay, whenever there is hemolysis, there will be excess stimulation of erythropoiesis, and sometimes the precursor form of the RBCs will come to the periphery. We'll discuss the precursor forms in the next section when we will discuss about the steps of erythropoiesis. And these precursor forms, they contain nucleus. So if you are finding nucleated RBCs in the peripheral smell, this is abnormal. That means somehow the precursor forms inside the bone marrow is coming to the periphery. It can happen whenever there is severe hemolysis, or it can also happen whenever there is some abnormal cells. For example, the cancer cells, they are infiltrating into the bone marrow and they're pushing these nucleated RBCs or the precursor forms of the RBC outside the bone marrow into the peripheral circulation. Okay, this is a higher magnification. You can see this is a nucleated RBC. Okay, so RBC they do not contain nucleus, so they cannot produce DNA and RNA. Okay, now they also do not have mitochondria. So RBC also lack mitochondria. So if they do not have mitochondria, they can only have anaerobic metabolism. Okay, they can only have anaerobic metabolism. And what is the end product of anaerobic metabolism? Anaerobic metabolism is your glycolysis. And what is the end product of glycolysis? You must be knowing that it is pyruvate. But if it is anaerobic metabolism, that means if you do not have mitochondria, this pyruvate cannot go for Krebs cycle, which happens inside the mitochondria. If pyruvate cannot go for Krebs cycle, it will be converted to lactic acid. So end product of anaerobic metabolism is lactic acid. This lactic acid will go to the liver. From the RBC, they will go to the liver. In the liver, this lactic acid will be converted to glucose through gluconeogenesis pathway. And this glucose will again come to the RBCs, where again they will undergo this glycolysis pathway and they will produce some ATPs. Okay, but you must be knowing that whenever there is anaerobic metabolism, they cannot produce enough ATPs. Okay, they cannot produce enough ATPs because ATPs are produced in electron transport cycle or oxidative phosphorylation, which happens inside the mitochondria. Okay, so ATP is produced inside the mitochondria. So if mitochondria is not there, they can have only anaerobic metabolism. So the amount of ATP is very less inside the RBCs. Okay, amount of ATP is very less inside the RBCs. Okay. Now you need to understand why nature has created this kind of abnormality inside the RBC. Why RBCs? Why they do not have mitochondria? Because what is the normal function of RBCs? The normal function of RBC is to carry the oxygen. Okay, normal function of RBC is to carry the oxygen from the lungs to the tissues. If they are carrying oxygen, they should not utilize this oxygen. I hope you are understanding. If they are carrying this oxygen, they are just carriers. they should not utilize the oxygen. If they're utilizing oxygen, they cannot efficiently carry the oxygen to the tissues. Okay, so that is why they will not use oxygen. They will not use oxygen. Okay, so if they have mitochondria, of course there will be some the mitochondrial pathways will be active, and the mitochondrial pathways, they require oxygen. Okay, inside the mitochondria we have this electron transport cycle or the oxidative phosphorylations, and all these processes they require oxygen for production of ATP. But RBC, because they are carriers, they should not use oxygen. Okay, so that is why they do not have mitochondria, and they just simply carry the oxygen. They cannot use the oxygen. Okay, I hope this concept is clear. So they do not have mitochondria, they will not use oxygen. They have only anaerobic metabolism, and the ATP production is less compared to other cells. Okay, and from this you can understand that if they do not have oxygen, if they do not have enough ATPs, so they okay, so if they do not have enough oxygen, sorry, I mean, if they cannot use oxygen, and if they do not have enough ATPs, they cannot produce. They cannot. I mean, they should not produce nucleic acid. They should not produce nucleic acid. I mean, the DNA and RNA. So that is why they do not have nucleus also. They do not have nucleus also. Okay. So I hope this concept is clear. Now I'll go to the next understanding. Now there are three important unique pathways that happen inside the RBCs. Okay. Now apart from glycolysis, what are the three important pathways? The three important pathways that RBCs use. Now, the first one is the hexose monophosphate shunt pathway. Hexose HMP pathway, or it is otherwise known as the pentose phosphate pathway. Hexose monophosphate shunt pathway or pentose phosphate pathway. Okay. Now, as you can see, this is the normal glycolysis process. But what happens in hexose monophosphate shunt pathway? In this pathway, the glucose 6-phosphate will be converted to 6-phosphogluconate through the enzyme G6PD, that is glucose 6-phosphate dehydrogenase. Okay, so glucose 6-phosphate will be converted to 6-phosphogluconate through the enzyme glucose 6-phosphate dehydrogenase. In this process, the NADP plus will be converted to NADPH. NADP plus will be converted to NADPH. Now, this NADPH is required to reduce the glutathione. So GSSG is the oxidized form of glutathione, and it will be reduced to glutathione through the enzyme glutathione reductase. So this NADPH is required to form reduced glutathione. Now GSH is reduced glutathione. This is reduced glutathione. Now why it is important? Now this reduced glutathione, this will convert the H2O2 to water. Will convert H2O2 to water. H2O2 is hydrogen peroxide, and if hydrogen peroxide is there inside the cell, it can form reactive oxygen radicals. And that will cause tissue damage. That will cause cellular damage. So this is actually protective. This reduced glutathione is actually protective. So from this you can understand if a patient has deficiency of glucose 6-phosphate dehydrogenase, there will be reduction of NADPH, and the reduced glutathione cannot be formed. And so that's why those RBCs who do not have glucose 6-phosphate dehydrogenase, they are very likely for oxidative damage. Okay, they're very likely for oxidative damage. More on this we'll discuss when we discuss about the G6PD deficiency anemias. Okay, so this is the first pathway that you have to remember. What is the second pathway? Second pathway is methemoglobin reductase pathway. Methemoglobin reductase pathway. Now, what is the significance of this pathway? Now, what do you understand by methemoglobin? So, what do you understand by methemoglobin? So you must be knowing that normally hemoglobin contains the ferrous form of iron. But if this iron is oxidized, if this ferrous form is oxidized, it will become ferric form. If the hemoglobin contains the ferric form of iron, this is known as the methemoglobin. Now, we do not want this methemoglobin. Why? Because this methemoglobin has high oxygen affinity. That means the RBCs, if they contain met hemoglobin, they will have higher oxygen affinity. If they have higher oxygen affinity, they will just carry the oxygen. They will not release the oxygen to the tissues. Okay. So the normal function of RBC cannot happen if they contain this met hemoglobin, because it will increase the oxygen affinity, and increased oxygen affinity will not allow the RBCs to release oxygen to the tissues. So that is why we do not want met hemoglobin in our RBCs. So that's why RBCs have a protective mechanism that is known as the met hemoglobin reductase pathway. What happens in this pathway? Here, the met hemoglobin can be converted back to the normal hemoglobin, which contain the ferrous form of iron. This happens by NADH, and this NADH is produced by this glyceraldehyde 3-phosphate dehydrogenase, and also here by this lactate dehydrogenase. So in these two enzymatic reactions, NADH is produced, and this NADH is important for methemoglobin reduction. Okay, so this is the second important pathway. The third important pathway is this one. The third important pathway is this one, which is known as Webering-Rappaport pathway. Okay, so this is Webering-Rappaport pathway. So what is this pathway? So as you can see here, there is 1,3-bisphosphoglycerate, which is intermediate product of during glycolysis. Sometimes this 1,3-BPG can be converted to 2,3-BPG, or sometimes known as 2,3-DPG, that is 2,3-bisphosphoglycerate or 2,3-diphosphoglycerate. And again, it can come into the glycolytic pathway through the phosphatase, and the glycolysis can continue. Now, RBC tend to produce this 2,3-BPG. They tend to produce this 2,3-BPG. 2,3-BPG through this Webering-Rappaport pathway, sometimes known as Webering-Rappaport shunt. Now, why they produce this? What is the function of 2,3-BPG? Now, 2,3-BPG, it has opposite function of methemoglobin. So I told you methemoglobin will increase the oxygen affinity, but 2,3-BPG, it will decrease, it will reduce the oxygen affinity. It reduces the oxygen affinity. Because it reduces oxygen affinity, it can cause oxygen release from the hemoglobin. It can cause oxygen release from the hemoglobin to the tissues. So this helps in oxygen release to the tissues. So that is why this RBCs also produce 2,3-BPG or 2,3-DPG. Okay, so these are the three important pathways that RBC use for our benefit. Now we'll see how we demonstrate this increased oxygen affinity and reduced oxygen affinity. This is demonstrated by the oxyhemoglobin dissociation curve. So this is, you all must have studied in physiology also, oxyhemoglobin dissociation curve. Okay, oxyhemoglobin dissociation curve. Now here in the y-axis, we have the hemoglobin oxygen saturation, and in the x-axis, we have the oxygen, partial pressure of the oxygen. Okay, so in the y-axis, we have hemoglobin oxygen saturation. In the x-axis, we have partial pressure of the oxygen. Now there is a concept of P50. So what is P50? As you can see here, P50 is the partial pressure of oxygen at which 50% of the hemoglobins are saturated. Hemoglobin is 50% saturated. Okay, so this is the partial pressure of oxygen. Partial pressure of oxygen at which hemoglobin is 50% saturated. 50% saturated. Okay, and normally the P50 is 27 millimeter of mercury. This is normally 27 millimeter of mercury. As you can see here, at 27 millimeter of mercury, the hemoglobin is 50% saturated. This is the normal P50. And the next important point you can appreciate here is the curve is sigmoid shaped. It is a sigmoid shaped curve. It is a sigmoid shaped curve. Why it is sigmoid? Whenever the oxygen saturation is less, whenever there is a deoxyhemoglobin, means oxygen saturation in the hemoglobin is less, the hemoglobin tend to stay in a tensed state. Okay, so whenever oxygen saturation is less, the hemoglobin tend to stay in a tensed state, tensed form. As you add more and more oxygen, as the process of oxygenation starts, this tensed form will gradually become a relaxed form. It will become a relaxed form. It will become a relaxed form. I hope it is clear. So if the oxygen content is, if the partial pressure of oxygen is less, the hemoglobin is less oxygenated, they will be in a tensed form. But as you add one molecule of oxygen, they will become slightly relaxed. As you add more oxygen, they will become more and more relaxed. Okay, so this is known as phenomenon of cooperativity. Cooperativity. Phenomenon of cooperativity. Okay, so this adding of oxygen, making it more relaxed, making it more relaxed. That means, uh, as you can see here, for example, this is a saturation of 20, sorry, partial pressure of 20, and here it is around 25% saturated. Here hemoglobin is 25% saturated. But if you increase the partial pressure of oxygen from 20 to 27, you can appreciate that there is a larger change in the hemoglobin saturation. There is a larger change in the hemoglobin saturation. So what this cooperativity does, once you add some oxygen, this hemoglobin will tend to take up more and more oxygen. They will tend to take up more and more oxygen because they have become relaxed. So because they are relaxed, they can take more and more oxygen, and the oxygen saturation increases rapidly. Okay, so change of, this is just an example, change of only 7 millimeter mercury of a partial pressure of oxygen is actually increasing 25% of oxygen saturation. So that is why the shape is sigmoid shape. Okay, that is why the shape is sigmoid shape. Okay. So you have understood the concept of tensed form, relaxed form, and the concept of cooperative. Because of cooperativity, they have a sigmoid curve. They have a sigmoid curve. Now we'll see what are the factors that can shift this curve towards right, and what are the factors that can shift the curve towards the left. Now, rightward shift means what? Whenever there is a rightward shift, it will reduce the oxygen affinity. It will reduce the oxygen affinity. Yes, and because of this reduced oxygen affinity, it can deliver more oxygen to the tissues. Okay, so whenever there is a rightward shift, okay. So as we are discussing, whenever there is a rightward shift, what do you understand by rightward shift? That means the curve is shifted like this, the curve is shifted like this. What is the meaning of this? Whenever there is a rightward shift, it means the oxygen affinity is reduced. That means there are more tensed form. Tense form has reduced oxygen affinity. As I have already told you, deoxyhemoglobins, they tend to be in a tensed form. Whenever it decreases the oxygen affinity, there will be increased oxygen delivery to the tissues. There will be increased oxygen delivery to the tissues. Now, what are the factors that can actually cause rightward shifting of the curve? Now, in conditions where we need more oxygen, those conditions can actually shift this curve towards right. So, what are those conditions? For example, whenever there is increased CO2, so at the cellular level, if there is increased carbon dioxide production, now the cells require more and more oxygen, and they want to throw out the carbon dioxide and take up more and more oxygen. So, in that condition, the curve will shift towards right. Okay, so I know that you must be knowing that whenever carbon dioxide concentration increases, it shifts the curve towards right and release the oxygen, and this process is known as Bohr effect. This is known as the Bohr effect. This we must have studied uh, in the physiology also. Similarly, whenever there is a decrease in pH, this you can understand like if a cell is having anaerobic metabolism, there will be production of lactic acid. Okay, the ultimate end product of glycolysis is lactic acid under anaerobic conditions. So this lactic acid will reduce the pH, it will become more acidotic. So that means the body will understand, the RBCs will understand that the cell is in a oxygen starvation state. So that's why they will try to give more and more oxygen to the tissues. So that is the reason whenever there is reduction in the pH, that will cause rightward shifting of the curve. Similarly, whenever there is increased temperature, that can also cause rightward shifting. And we have already discussed that when there is 2-3 BPG, that can also cause rightward shifting. So rightward shifting means reduction in oxygen affinity of the hemoglobins and increased oxygen delivery to the tissues. Okay. Similarly, if you see what are the factors that will cause leftward shift, leftward shift means it is exactly opposite of what you have discussed for rightward shift. Left shift means the oxygen affinity of the hemoglobins will increase. There is increased oxygen affinity, and they will not release oxygen to the tissues. They will not release oxygen to the tissues. So examples of this, opposite of what I have discussed here. So whenever there is decreased CO2, or whenever there is increased pH, or whenever there is decreased temperature. So all these factors can cause leftward shifting of the body. Also decreased Q3 BPG. These points already know. Apart from this, you have to understand that there is a type of hemoglobin, that is hemoglobin F, or fetal hemoglobin. Fetal hemoglobin. This has very high affinity for the oxygen. And that is how it can actually bring oxygen from mother blood to the fetus. That's why hemoglobin F has higher oxygen affinity. So this will also cause leftward shifting. Apart from that, we also discussed that met hemoglobin will also shift the cord to left. This also has higher affinity. Okay. So these are all the points about the oxygen affinity. Now coming to the last concept, that is the fate of senescent RBCs. So what is the normal lifespan of RBCs? Normal lifespan of RBCs is around 120 days. It is around 120 days. So as the RBC near the as the RBCs become more and more older, and towards the final days of his towards the final days of its life, this RBCs will have some age-related changes. They will have some age-related changes. They'll have some age-related changes. Now, because of these age-related changes, they cannot negotiate through the splenic cords of Billroth. These are the splenic cords of Billroth, and this is the splenic sinusoid. This is the splenic sinusoid. So normally what happens whenever RBCs pass through the spleen, especially the open circulation, they will go into the splenic cords of Billroth. From the splenic cords of Billroth, they have to go to the sinusoids through narrow fenestrations. They have to go to the sinusoids, and from the sinusoids they will go out of the spleen through the splenic vein. So that is how normal circulation of RBC happens inside the spleen, especially in the open circulation. Details of this we will discuss when we will discuss more about the spleen. Okay, this is just a schematic diagram to understand it better. So this is what normally happens. But what happens whenever the RBC has these age-related changes? That means RBC is nearing its death. Okay. So these age-related changes will make RBCs less deformable. So this will make it less deformable. It's less deformable. As it is less deformable, they cannot go from splenic cords to the sinusoids. It cannot negotiate from the splenic cords of Billroth to the sinusoids, and they will get stuck inside the cord. They will get stuck there. And if they get stuck there, it can be easily identified by the splenic macrophages, and they will engulf the RBCs. They will engulf or they will destroy the RBCs. Okay. So this is how the normal RBCs gets destroyed after 120 days. This process also happens in liver and also in bone marrow. So what is the end product? Now this RBCs, they contain hemoglobin. Hemoglobin will be converted to heme. It will be converted to heme and globin. So globin is a polypeptide, so it will be dissociated into amino acids, will go into circulation. But the heme will be converted to bilirubin. Heme will be metabolized to bilirubin. Now this bilirubin, through the circulation, through the portal vein, it will go out to the liver by binding to a protein that is known as albumin. Okay. So by binding to albumin, it will go to the liver. Inside the liver, it will be conjugated with glucuronide. And after conjugation, they can go into the bile. And gets excreted. This bilirubin metabolism also we'll discuss in a separate video. So this is what normally happens. So after 120 days, there will be age-related changes, and so that's why the RBCs, they cannot negotiate from the cords of Billroth to the sinusoids. The splenic macrophages will identify this. They will cause metabolism. They will cause damage to the RBCs. This hemoglobin, the hemoglobin inside the RBCs will be converted to heme and globin. Globin will be dissociated into amino acids, which will go into the blood, and the heme components will be converted to bilirubin. This bilirubin, by binding to albumin, will go to the liver. Inside the liver, again the albumin will be dissociated, and this bilirubin will be conjugated to glutamate, and this conjugated bilirubin will go into the bile, or biliary system. Okay, so these are the basic concepts. These are the properties of mature RBCs. In the next videos, we will discuss about the process of erythropoiesis and its regulation.